Hello everyone, and welcome to the second day of Physics Meets Biology. So I'm Ruhi, I'm the president of IAPS, the International Association of Physics Students, and I'll be the host today. So as you can see on the screen, here is our schedule for today. We'll be having 3 really interesting talks. And first we'll start off with a brief introduction into IAPS and IUPAB. So we've already done this yesterday, so we'll keep it brief today. Okay, so I'll get started with that. So IAPS, the International Association of Physics Students, is a nonprofit, and it's based in France. And you can see the rough spread of our members around the world. So if you'd like to become a member, you can join a national or local committee in your country. And if there isn't a national or local committee, you can join as an individual member. Here are some of our members, and anyone is welcome to join as long as you're a student of physics or a related subject at university level, or have recently graduated within the last 12 months, sorry. Okay, so some of our activities include the International Conference for Physics Students. So this is held in different countries each year. This year will be in Mexico, but online. The year after will be in the Philippines. So it'll be great to see some of you there. We have Planks, which is our main competition. This is the physics league across numerous countries for kick-ass students. So the winners of each national preliminary competition go on to the international finals, and these will be held in Munich next week, which is quite exciting. We have a bunch of other things going on. GIAPS is our journal, and here's the general spread of events that we have throughout the year. So these are the ones that happen every year. This year, we have some special things as well. For example, ScienCon was just recently held in Poland. We have, we're on the International Steering Committee of the International Day of Flight, which will be held next month, which we're looking forward to. Lots of things going to happen there. The EPS Forum is hosting a large delegation of IAPS members, so they're getting funded accommodation and travel and everything to attend, so that's another thing happening. And of course, this year is the International Year of Basic Sciences for Sustainable Development, as proclaimed by the UN, and we're on the International Advisory Committee for that. So there's lots of things, other things going on, and we give grants. And one way to get more involved is to join working groups. You can see all of this information on our website. Social media will tell you a lot more than I've just told you now, so check us out, and thank you for listening. I'll pass over to IUPAB if they're here. Okay, if not, we might do the IUPAB presentation at the end. So let's start the first talk. So Sara.Koester, great to have you here. I'll pass over to you.
Okay, yeah, thank you very much for the invitation to be part of this. It's been great already yesterday, and really looking forward also to the questions and discussions afterwards. Yeah, I'm Sara Kuster. I'm a professor for experimental biophysics. Oops, sorry, this is too fast. I'm looking for my laser pointer, actually. Here we go. I'm a professor for experimental biophysics at the University of Göttingen in Germany. That's actually the same city where Helmut Krupmüller is from, who will speak tomorrow. That's a coincidence, I think, in choosing both of us to speak here. Yeah, I would like to show you a little bit of how we study biological systems using physical methods. And I guess that many of you have attended already yesterday, or have in any other way been in touch with biophysics one way or the other. And that's why I would like to start with a little experiment together with you. I've never done this with such a large crowd, but let's see if it works. And I would like to build up a word cloud of terms that come to your mind when you think of physics and biology. So please either go to vote.ac or use your smartphone and scan this QR code, or yeah, maybe you even have an app on your cell phone that's called EduVote. Coincidentally, then you can also use that. Go there, and I will start this now. And now you can type in the ID Köster in some kind of field, then choose text and provide one keyword that comes to your mind when you think of physics and biology. And everyone can participate on Zoom, on YouTube. As I said, it seems to work. One person has already submitted something, so that's a good sign. So we'll wait a little bit for you to put in your ideas and see what comes out of that. I really hope that the quality of the QR code is good enough on YouTube as well. Otherwise, just go to this website, vote.ec. That should work as well. And maybe you remember something from Tony Watt's talk yesterday, or any other encounter you had with biological physics, biophysics, any of that. Okay, it's going very slowly. Not sure if this is a technical issue or people just being, oh, by the way, it's completely anonymous, of course. I will not know or see anything that you put in. So, okay, let's give it just like 1 more minute or so, because we want to learn about some other things as well. Of course, it works best if many people take part. Okay, this seems to be a not so active crowd. That's a pity. I hope it's not just a technical problem that keeps you from participating. That would be sad, and I would be sorry about that. Give you a few more seconds. Okay, I think I'll stop it here. 14 participants, 15. Okay, and let's see what you came up with. This normally takes a little while. Okay, that's very nice. Proteins, life cell, biophysics, complex systems. Passion, I like that one. Fundamental science, yes, that's true. Interdisciplinarity, that's also true. Mechanics, DNA, proteins, AFM. Very nice, cool, thank you very much. Okay, so yeah, I guess some of these keywords we will actually touch upon during my presentation, and then maybe some more during the discussion. So actually, what is physics of cells, which is actually the major topic that we are studying in our group? When you look at your own body and you look at the different cell types that you have in your body, let's say the brain cells, the neurons in your brain, these are the neurons, I guess. No, these are skin cells, I think. The neurons, the muscle cells in your heart, the red blood cells, the connective tissue cells, whatever you can come up with. You realize that in your body, you have about 200 different types of cells. And that's actually quite amazing because they all are built up from more or less the same building blocks, but they have very, very different functions. And they also have very different mechanical requirements. And we see that when we, for example, compare muscle cells that are actually contracting and relaxing over and over again. Very differently when you look at brain cells. They are protected by the bone, but they are experiencing hydrostatic pressure and compression. Skin cells in the epithelium have to withstand shears, or you have immune cells and fibroblasts. Those are two cell types that migrate a lot. And when you think about that, it's clear that they all have to somehow have different mechanical functions and mechanical properties. And where do these mechanical properties come from? Well, that's actually mostly determined by the so-called cytoskeleton. And when you translate that word into English, it would say something like skeleton of the cell. And so the skeleton of the cell is, of course, not made of bones, but it's made actually of biopolymers. And by polymers, we here just mean molecules that are very, very long and very thin. So the diameter is like, I don't know, in the order of 10 nanometers, and the length is on the order of several micrometers. So that's orders of magnitude different. And then when you look at these different biopolymers, here's a picture of a cell that we took with fluorescence microscopy, where we can actually label certain components of the cell in certain different colors. Here we labeled the actin filaments in red, the microtubules in blue, and the intermediate filaments in green. These are just different types of filaments that we find in the cytoskeleton. And they together basically form this cytoskeleton. Interestingly, they have very, very different mechanical properties. And by mechanical properties, I mean how easy it is to bend them. That is given by the so-called persistence length. how easy it is to stretch them. And in that way, the three of them are very different. And now when you put them together in this network and you add crosslinkers and so-called molecular motors, which are little active molecules that consume energy and then lead to directed motion. You also have electrostatic and hydrophobic interactions. So you can already see where all the physics is here. Then you get this very, very complex composite network. And now, of course, the next question is, why would anyone be interested in understanding that or studying that? And that is because, first of all, we're physicists, so we like fundamental research. And in biological or soft matter physics, it's just very interesting to understand what's going on here. But this actually goes beyond this, because also for material science, it's very interesting to understand these biomaterials, because sometimes they have properties that you don't find so easily in a synthetic system. So some of these networks or filaments have properties that very difficult to make in a synthetic chemical system. And so it's good to learn from nature how nature actually comes up with these interesting properties. And there's a whole fairly new community of people who are working on a so-called synthetic cell, and they're trying to engineer and build up a cell from scratch. And by that, they mean something that is able to divide or grow or has any other properties that a cell has. And that's actually at the moment still at its beginning. But I guess your generation will be the one that will actually bring this much further, because it's really a very new field, subfield of biophysics. And then last but not least, of course, it's also interesting to understand from a medical point of view how this works, because you could imagine that, let's, for example, assume a muscle cell, and then one of these network types doesn't function properly. And that's why this muscle cell doesn't contract well. That could lead to a heart disease, for example. And understanding where this comes from is the first step in trying to cure the disease and to understand what can be done about it. So that's basically why we study it. Today, by the way, I will focus mostly on these intermediate filaments, which you will see later, because that's mostly our focus of study. But the other two are also super interesting and important, and we will also speak a little bit about those. The other question now is, how can a physicist, and as you probably all know, physicists like to simplify things and find simple models and try to come up with something that they can understand and calculate and put in a model and find some numbers and measure something. How can we still study something as complex as the cytoskeleton that's really very complicated? And there are actually two major ways of approaching that. One is called bottom up, where you start with a simple system and you build it up all the way until you have something that's more complex. The other one is called top down, where you start with a more complicated system and you simplify it more and more and more until you can actually understand certain aspects. And so how do we do that? Well, we could, for example, study living cells, like the picture that I showed you before, but we can also simplify the system by saying, okay, let's not study the whole cell. Let's study a so-called in vitro system. In vitro means in glass, and that means in a reaction tube or something. And that means that we take certain proteins out of the cell. In this case, it's just two different types of proteins in two different colors shown here. And we put them together and we study just the interaction between the two, without all the other stuff around it, which makes our life much simpler. We can also study just one component. That's also an option. And then we simplify the system even more. And we can, for example, say, okay, this is a network, but let's study the most simple network that we can think of. And the most simple network is just two filaments that we study and we look at how they interact with each other. Yeah, so that could be a very simple system. But we could also say, let's just study one of these polymers. Let's see how it bends. And let's see how we can stretch it. Yeah, so we have a really simple system and we only look at the properties of this one polymer. And as you can see here, we're actually going down with the scales. We're going from a full cell down to the network level, down to the filament level. And then we can go inside the filament and try to understand what's inside the filament. Then the methods that Tony showed you yesterday actually come into play, and that's very important. And the modeling that Helmut will show you tomorrow will also be very important for this kind of level of hierarchy that we have here. So experimentally, we have lots of methods at hand that we can use here. These are just a few examples. So for example, imaging, atomic force microscope, which is something that's one of you actually put on our disjoint word cloud in the beginning. Rheology, where we shear the material and we look at the response and see how elastic it is and how viscous it is. We can study deformation by stretching the cells, for example. Rheology again, we can measure forces, we can look at elongation, we can look at bending, stretching, and so on. And you may now wonder how we actually do that, because all of this is very small. You see the scale bars here. This is 10 microns, 10 microns. Yeah, so the length scales are very small. How can we still stretch or bend something like that? And I will show you some examples of that. And then we try to complement this by some simple physics models to really try and understand what's going on the molecular level, and then feed this back into these higher order levels. What I would like to do today is first look at how the filament architecture, this would be this level, is related to the filament mechanics. Then we will go to the network level here and look at that for a moment. And then we will go to cells in the very end. Yeah, so we will basically go backwards now in this row. So let me start with a very old experiment. This was done by a colleague of ours, Paul Janmi, in 1991. That's been a while. And what he did is he did so-called bulk rheology, which means that he made a network out of these different biopolymers, actin, vimentin, microtubules. These are the three that you know from inside the cell. He also used fibrin, which is a protein that you find in blood, and that's important for blood clotting. And he looked at those in a rheometer, which means he put it between two plates, and then he sheared one of the plates and he looked at the response. And what he could see when he plotted the strain, which is basically how much he deformed it, against the stress, which is how much force he needed to use it. And you can see that it's an old paper because the units are very old-fashioned here, no SI units. What he could see is that microtubule and actin networks ruptured right away at very small strains and stresses, whereas the intermediate filament vimentin network remained undamaged, as well as the fibrin network. So he already could tell that these intermediate filaments, we abbreviate this with IFs normally, are very stable against stress. And this experiment was followed up upon much later by Laurent Kaplan, who did an AFM, atomic force microscopy experiment, where he put a filament on a substrate like this. Then he took this little cantilever from the atomic force microscope, and it's really like a little tip that he would pull through the filament until it ruptured. And now he could measure the original length, that's just this connection here, and he could measure the length just before it ruptured. And what he learned from that is that you can extend these filaments up to 300% before they rupture. That's actually quite amazing. That's really an amazing material. Most materials that you can think of, you can't extend that far. People then also did wide angle X-ray scattering, and this goes into the direction of what you saw yesterday, where they could actually see the signature of a so-called alpha helix in the protein structure. That's a certain structure that you find in proteins very often. And then after pulling, they could see a beta sheet, which is a different structure. So upon stretching, it doesn't just get longer, but something happens. And this is also what you can see in simulations. This is from the Bühler group at MIT, where they simulated these alpha helices and they pulled on the alpha helices, and then you get this beta sheet after a while. Yeah. So that's a very interesting series of experiments. And what we learned from this already is that intermediate filaments are extremely stretchable, actually. Yeah. And we recently followed up on these experiments, and we really like this experiment a lot. What we don't like so much about it is that you always get this interaction with the substrate. So it's always sticking to the substrate, and then you do something to the filament. So that's very difficult to model. So instead, we use the setup that looks like this. It's a combination of so-called optical traps. Optical traps are basically focused laser beams, here shown by the red cones, with which you can capture such a microsphere. The sphere is about 4 to 5 microns in size. And we bind to the sphere a filament, and we bind the filament to another sphere. And we have 2 of these traps. I will show you in a movie in a minute what we can do with that. And we combine this with a fluorescence microscope, which just helps us to see all of this. And we combine it with a microfluidic setup, which helps us to control which kind of liquid is here, which kind of buffer, which kind of salt concentration, pH, and so on. We can really play with that. So this combined instrument helped us to do the following experiment. Here's a little animation that we prepared for that, where we have this microfluidic channel and you have 3 laminar sub-channels. There are no real walls. This is just for a guide up to the eye. And then with the optical traps, we first capture 2 of the beads. Then we move to the filaments and we wait until the filament is attached to them, lots of other stuff as well. Now you see a real movie from our microscope and you see how the filament is attached here to this one bead, and there's also some other stuff that we would actually like to get rid of. There's also some stuff here that we don't like. And then Johanna here had to be a little patient and go back and forth and wait until this, now you have really just one filament. And then what she can do is she can move this bead and by that stretch this filament. So that's why these are called optical traps, but also sometimes optical tweezers, because you see how small all of this is, but you can use the laser beam to capture the bead, and then like with tweezers, you can move it around. And at the same time, you can use this bead actually to measure a force, because physically this trap works like a Hookean spring. So it has a stiffness. We can measure the stiffness beforehand. And by measuring how much the bead is moved out of the center of the trap and knowing the stiffness, we know the force that's acting on this trap. So we can measure the extension that we apply here, and we can measure the force. And actually, we measure the extension with a nanometer precision, and we measure the force of the piconewton precision. So it's all very small, and it's all exactly the units that we need to study biophysics. And then what do we get? It's something like this, where we plot the force against the strain. And the strain is actually the normalized extension. So instead of plotting the extension, we plot the extension divided by the original length. Why do we do that? Because then we can compare different filaments with each other, even though they have all the different lengths. And these are the typical curves where we see a trace and a retrace. And let me show you 3 really important aspects of this curve. The first one is we have different regimes. First, it's elastically extending. So the force is proportional to the strain. This is what you learned in your freshman physics class, basically. And we really see this here in the system. We can get a spring constant from that. We learned something about the filament. Then we have this region where we see almost like a plateau. So we need very little force to extend it very far. And then when we pull even further towards a strain of 1, and now a strain of 1 corresponds to twice the original length. Yeah? So it's already very much extended. Then it gets stiffer again. So that's the first important point. The next point is that we have this hysteresis, which you also know from your physics studies what that is. It's basically the area between these 2 curves. And we can look at that area because that's basically giving us an energy. And we can compare the energy that we put in, that's the area under this curve, and the area that we get out. And then we learn something about the dissipated energy. And you see here that the dissipated energy in this case is between 70 and 80%. So more than half of the energy that we put in is dissipated. And that's why we and some other people believe that these type of proteins are actually like a shock absorber for the cell. They can take up a lot of energy, which really helps the cell to survive in certain situations and to perform its function. And the third thing is a little bit more subtle, and that actually concerns this point here, because after going all the way forth and back, you end up at the same point again. And that means that we have no plastic deformation that's visible on these length scales here. Yeah, so we really got back here, and that's quite interesting. So now we started to work with this kind of system. And what we first did is we actually used different pulling speeds, loading rates, that's the same thing, between 0.05 micrometers per second and almost 5 micrometers per second, so going over 2 orders of magnitude. And what you see is that when you pull very slowly, you can pull very far beyond the 1 here and even further. And some of the individual filaments in light blue here go even up to 1.5. So that's 2.5 times the original length. When you pull fast, this doesn't work. Then it stiffens much earlier. And so another group, not us, actually termed this the safety belt of the cell, because when you think of your car and you pull on your seat belt, then you actually can pull it very far when you pull slowly, but when you pull fast, it actually blocks and you can't pull that far. So that's actually, I think, quite a nice picture. And it could actually be very relevant for the cell, because cells are this kind of cool material that has to be flexible when they have to squeeze through constrictions, or they have to move somewhere and they have to be motile, but they also have to be rigid and persistent when there is a force impact. They can't just fall apart. And the combination of these two properties in one material, that's actually what makes a cell a really interesting mechanical object. So the experiment are these colorful lines here. And we actually also did some modeling, and we did actually a very simple model. I'm not going through the math with you now, but of course we did all that exercise. We combined a spring, just a hook-in spring, with a polymer model, for example, a freely jointed chain. And we also included in our model that every element of our filament can either be in the alpha helical state, or it can open up and become something longer. It could be a beta sheet, could be something else. And this basically we took from the literature, because people had hypothesized or shown in experiments this before. And so we combined like this two-state model here with this polymer and spring in series. And when you calculate that, you actually get the curves that you see here. And we can fit these curves very nicely to our data. And then we actually learn something about our system. And actually these zigzags here are just an artifact from the averaging. So they're not real. They're this problem with the averaging, which is basically because if one filament stops here, then the average jumps down a little bit, but that's not really a problem. Yeah, and the next thing we did is not just going up and then waiting until the filament ruptured, but we went back and we stretched again, and we went back and we stretched again. So we went in cycles basically. And here you can see a cycle where we went to a larger distance every time. And you see again the force against the strain like before. Black is early times and red is late times. And what you see is that with every cycle, the filament becomes softer. Why do I say that? Because the original slope here in the beginning becomes less steep. And less steep in a force versus strain diagram means that your spring constant that you measure is smaller, so it's softer. So what we see is that there's some kind of dependence on the strain history, what has happened before. And we see the filaments get softer, and interestingly, we still see no plastic deformation. So we still end up here at the 0.00 point. And this is something that we can't really understand with our simple model from the slide before. So what we did instead is we went back to the molecular structure of the filament. So we really looked at how are the monomers actually arranged in such a filament. And this is here just a model of a piece of the filament where you have so this is actually one monomer, and there's another monomer. So this is a full dimer here. Then we have a tetramer when we put this red thing together. And 8 of these tetramers make up the filament, but not the whole filament, but only this short piece. And then many of those you put in series behind each other, and then you get the full filament. So it's a very complicated and actually hierarchical arrangement of these structures. And we used exactly this arrangement and set up a Monte Carlo simulation. So in our Monte Carlo simulation, we put together elements that have a spring with something that can open and close in series. This is our two-state model, basically, alpha state, beta state. And we put them all in parallel. Yeah, we coupled them actually here. And then we have some other springs that we need because we put these elements behind each other. And here you can basically see the full simulated filament. And this was a collaboration with our colleague Andreas Jansow from physical chemistry. And here you actually see very clearly the interdisciplinarity that's so important for us, and his former PhD student Hannes. So what we get when we actually use this model is really our experimental observation. We can actually pull and relax, and interestingly, we get back to the same length, but it gets softer. Why is that? This is because the length of this element is set by the shortest element. And the shortest element is that maybe only one alpha helix that's available. All the other ones might be in beta state. It's still in the short length. So that means the following. We start, we pull, and then they open up one after the other. Yeah. And when all of them are open, then we elongate. And this is what we see in our experiment. Now, when we go back and we relax our pulling in the optical tweezer, they will close, but not all of them, and not all of them at the same time. But as soon as one of them is closed, we're in the short situation. And then when we pull again, we will actually have a softer filament because we only have to open a few of them. The other ones are already open. But we get back to the same length. So this is a little bit subtle, but I think you probably understand what I mean. Otherwise, we can discuss this further later on. Looking at that, which describes our experiments really well, the question, of course, came up, what happens if we wait longer for all of them to close? Will the filament then get stiffer again? And this is what another student, Julia, then tried. She pulled, she relaxed, and then she waited. And then she pulled and relaxed. And her question was whether this pulling here was the same stiffness as this pulling or not. Here's the experiment from before. The first cycle in red, and the blue one right after, without waiting, is softer. When she waits 30 minutes or 60 minutes, we have the same situation. So interestingly, we don't see that there's recovery even after 60 minutes, which is actually a long time scale for a cell. And that means that this two-state model that I showed you does actually not describe the behavior that we were actually looking for. That's why we had to come up with a more complicated model that describes the tensile memory, that there's no plastic deformation and no recovery. And that's where we are right now. Yeah, so this is basically our state right now. Not the end of the story, of course, because we haven't measured this model. We just postulated because it fits our data. What you see here is the model from before along this coordinate. You have the alpha helix here, an energy barrier, and the beta sheet. But we add a hidden reaction coordinate. And when you look in that direction, you can see that the alpha helix can actually unfold into random coils. Those can be shorter or longer, and we can also shuffle between them. And sometimes it also goes into the beta state, but then it actually stays there. And this is what people have seen in their X-ray experiments, for example. So this is how we can explain the data at the moment. But of course, as I said, this is probably not the end of the story. We're still thinking about this and working on this. And now I would like to come to the next topic that I announced to you, disentangling network mechanics. So now going from the single filaments that we were looking at here, especially this experiment, taking into account the molecular models that we have, we now move further up in hierarchy or in length scale and look at networks. And here I would like to show you a very simple experiment because I think it's beautiful because it's simple, but it still tells us so much about a network system. This was done by Anna Schepers, who prepared a network between two glass lines, and she included in that network these little beads. Yeah, these are beads of, let's say, 1-2 micron size. And now she looked at traces of these beads over time. And then she plotted the so-called mean squared displacement, where you look at how far does the bead move within a certain time, but you take the square of that distance, and then you average over all those distances, and you plot it against the time that you give the bead to move. When you have just diffusion, you just put these beads in a water environment, then you will get something like this. So you will get just a straight line. When you have a confined bead in a network like this one, you will see something like this. And that's exactly what she did. She started out with, oh, maybe I should have mentioned the axes here are logarithmic. That's important. That's when you get these pictures that I just showed you. So she started with this network of momentum filaments, and after 3 hours, she took the first data, and you see that it's actually almost diffusive. That looks actually almost like no network is there. But when she now waits longer, and she actually was very patient, she didn't just work for 1 day, but then she continued the next day, and even after 1 week, she took data. And what you then see is that with time, the network matures. And actually, this is the point where you can say, now we have a network, because now the mean square displacement is smaller than the bead diameter. Yeah. Here you see a picture of the network. You see that it's actually very difficult to image these networks because they're fluctuating like hell. But indirectly, we can image them because we can look at how these beads move, and you see how much confined the bead is. Yeah? It can really only move within this very small circle. So this was the first step, and this is basically the idea of the idea we have of these full networks. And then Anna said, okay, let's now look at what's actually inside the network. We have already characterized the filaments themselves. We have pulled on them, but when we pull on a network, actually something else happens. We also pull on the interactions between two filaments. And this is what you see here in this pictogram, but you also see it here in this animation. We have two filaments now with 4 optical traps, and we bring them in close contact. And then, so Anna always says we're playing the nanometer violin here. We're using one as the bow and pull it down, and thereby deform the string. And you can see that here very nicely. Yeah. And by the deformation, you can actually already see that there is an interaction. So we can measure this interaction because we're using these optical tweezers with which we can measure these piconewton forces. And this is exactly what you see here. So here's again that animation just spread out in individual pictures. So what we see is that starting at a force of 0, the force increases up to about 50 piconewton. And then at some point between these two pictures, the force ruptures. So this is our maximum force. Yeah, this is the force that we measure. And of course, we don't just do this experiment once, but we do it many, many, many times. So we can plot a histogram, which you can see here. And in this histogram, you see also a simulation that we added here. This is our control. And now we said, okay, let's think about what kind of interactions could be there. We really only have the protein. We have no binding partners or anything like that. So we could have either electrostatic interactions because there are charges involved, or we could have hydrophobic interactions because the proteins have hydrophobic groups. So the first thing is we decrease the hydrophobicity by adding a detergent that's added here in 2 different concentrations. And you see that not so much happens, like the histograms look very much alike. So that's apparently not the important point here. And keep that in mind, because I will show you a different example later on where it does actually matter. And then we increased electrostatics by adding magnesium. So we added divalent ions that kind of glue together the filaments more strongly. And you see that this really works. So even with 5 and then with 20 millimolar magnesium, we get histograms that are spread out towards higher forces up to 200 piconewton. 200 piconewton is actually already quite high for a biological system. And we can, with some modeling, as I told you, this is here the simulation, we can also get the force-independent binding rate of our system. And then you see that actually, interestingly, not at 5 millimolar magnesium yet, but at 20 millimolar magnesium, we can see that this really increases. And now what's really interesting is that then I went back 10 years in my science career. And at that time, 10 years ago, we had actually published a paper together with a PhD student at that time, where we had looked at vimentin networks in little droplets. So this is a little bit into the direction of the synthetic cell idea. And we added different amounts of magnesium, and we could see that between 6.5 and 12.5. And actually, if we do it more accurately at about 10 millimolar, we get this collapse of the networks. So 10 years ago, we already saw that there is this interaction between these filaments that's very strong. But only now, 10 years later, we have the technical methods and possibility to actually measure and quantify the forces as you can see it here. Yeah, so this is, I think, a good example where you see how methods development and advancement in not just the science, but also the observation methods really helps us to learn more. And I think it's really nice how this still fits together. At the time, it was much rougher, and now it's much more quantitative. Yeah, and last but not least, let's move to composite networks. So now moving from these single networks that Anna studied with microbiology, but then also with these optical tweezers, let's move to mixed networks and then also look at mixed networks in cells. So here, Lotta and Laura did an experiment together, which is very similar to what I just showed you from Anna. With 4 optical traps, and this animation was actually done by Markus Osterhoff. 4 optical traps, we capture 4 beads now, and then we use 2 traps to capture a vimentin filament, like before. We use the other 2 traps to capture one of these microtubules. I hope that the movies get transferred well in YouTube. And then we bring them in contact. And now we move one of the filaments over the other one, and now they're not in contact yet, and they're not really interacting, but at some point you will see a little flash just in the animation, of course. Now you see a little flash, now it's interacting, and then we pull this together, and again, we can measure these forces that are existing here. And again, just two types of protein, nothing else in the system. And so, yeah, this is basically the experiment. Here's again just a few snapshots from the real experiment. We do exactly the same thing. We measure the force just before it ruptures, like between these two frames here. And this is the force that we have here. And what we then see is that we get this distribution of braking forces again, and we do what we did before. We added magnesium chloride, so electrostatic, and then you see that the forces really increase to very high forces again. They're rare, these events, but high forces. So there is some kind of electrostatic contribution. We also added our surfactant or detergent, and then you see that actually goes down. So in this case, for this particular example, we could actually show that as electrostatic and hydrophobic contributions, which is also very interesting for the system. But of course, as I said, it's really a very simple system. This is this in vitro approach that I mentioned in the beginning, where we don't look at the whole cell, but we actually really take it out of the cell and work with just these proteins. We also do some cell experiments, and that's the work of Anna Blob and David Wenske that I would like to show you now, where they study these fibroblast cells. Fibroblasts are connective tissue cells that are very migratory and very nice cell system to study. And Anna puts them on these protein patterns that she prints actually with a certain method. She makes all these little anchor patterns here. And why does she do that? Because she wants the cells to be comparable. So she forces them into a certain shape that you can actually see here. That is good for statistics, because David is actually very interested in mathematics and data analysis. And so he would like to have lots of data that he can then compare. The cells are also very flat on these patterns because we give them space where they can spread out, and then they really try to cover this. And with this kind of pattern, this particular kind of pattern, we enforce polarization, because a cell now sitting like this has a leading edge here. This would be the direction of motion, and a rear end. Of course, we can then compare this to non-polarized cells like circles. Yeah, and here you already see an example of not just the pattern that's in blue underneath, and then you see a microtubule network inside the cell in green. And you can already see that there is already some kind of difference between these two types of patterns here. So why are we interested in these microtubule patterns? That's actually the filament that I showed you in the last example before with the optical traps as well. Because microtubules are very dynamic. When you look at a movie like this one, this is about 10 times real time that I show you here. You see that there's lots of stuff going on. For example, we have these so-called buckling events. So this can be a straight microtubule, and then actually it's not known yet whether it's compressive forces pushing from here and from here, we get this buckling, or is it a lateral force that pushes from here, and that's why it buckles. So this is one open question. So what we see is we see buckling, but we also see microtubules grow and shrink because they're very actively, dynamically growing and shrinking all the time. And what we would like to understand now is, how does the Vimentin network that you see here in red now influence this microtubule network? Yeah, does it at all influence it? So we're currently working with fixed static cells, and then in the near future, we'll start working on living dynamic cells here. So what you basically see is that this is quite boring when you just look at the different cell types, the different patterns, the crossbows and the circles looks almost the same. We also looked at different cell types. This is a cell type with Vimentin. Vimentin KO means knockout, so there's no Vimentin in there. For comparison, that's also very boring. So there's really no big difference. But then when we look at the cell, we realize that the Vimentin is not homogeneously distributed through the cell. It's actually more like in the center around the nucleus. There's not so much Vimentin out here. So the next thing that David and Anna did is they call this to onionize the cell. I really like that expression. They actually look at the cell, and then they look at different regions. Yeah, just going from the edge, they go to the center, and then they define these different regions. So the cell center would be here where there's lots of Vimentin. The cell edge would be here where there's very little Vimentin. And when you then look at these curves, and now we're actually interested in these reddish curves, so those are the cells with vimentin, you see that in the center with lots of vimentin, the average curvature of the microtubules goes up. Yeah? And we can also plot the curvature against the vimentin intensity, then it's actually much clearer. And then you see where we have lots of vimentin, we have strongly bent microtubules. Yeah? And so this tells us probably that there is an influence, and this is what we're looking at the moment. All right, so with this, I would like to summarize a little bit what I told you. So I hope I could convince you that cells have physical properties and that physicists have tools to study them. And nowadays, there are more and more really cool tools coming up, and I think that's already a reason to study biophysics, actually. I then moved to the cytoskeleton, which determines the mechanics of the cells. That's actually what I and my group are working on mostly. And we focus a lot on intermediate filaments because they're a really interesting material. They're highly extensible, which makes them a high performance biomaterial. So you could call them a safety belt, a shock absorber for the cell. There are actually other biomaterials with similarly cool properties like silk, for example. And it's really interesting to see what nature came up with and how we could maybe copy this for synthetic materials. And another thing that I aimed to show you was that there's really a hierarchy of length scales. When you look at cell mechanics or network mechanics, you have to take into account how do the filaments behave, how do the filaments bend, how do the filaments interact with each other, and then you can put it all together in a way. The big open question that we all have is how do we bring the bottom-up and the top-down approach together in a smart way? And are the in vitro results really relevant for the cell? And can we as physicists quantify and understand what's going on in a cell with our simple models that we can understand fairly well already? And the last thing I would like to say is that I'm really thankful for my research group in Göttingen. And this is a fairly old picture. We really have to take a new one now. They're really great people and really enthusiastic people with great ideas, and they're really fun to work with them. And the other thing that's really important for us is to have collaborators from all kinds of fields. So for example, biologists, biochemists, chemists, X-ray people, optics people, and bringing all that knowledge together and being very open and exchanging ideas and discussing ideas with each other is actually very important here. And then of course funding is also important. Without that it wouldn't work because all these PhD students need a salary. And now I'm at the end of my talk, and I would like to thank you for your attention, and I'm looking forward to your questions.
Thank you so much for your talk. It was so interesting. So since we're a bit over time, we'll just go really quick with the questions. Okay, first one about the technique of selective opening in the Monte Carlo simulation. Is it based on how it happens in nature, or is it just a useful tool for simulation?
Well, what happens in nature is probably closest to what you can see in these simulations. So when you pull on a alpha helix or coiled coil, actually two alpha helices coiled together, then this will actually open up and form this beta sheet. You can coarse grain that, and instead of simulating every single H bond in that system, you just say, okay, we have a short and a long state, and we shuffle between the two. So it is. I think it is very close to nature, but it is a coarse grain model. I think that tomorrow Helmut Grubmüller will show you much more well-resolved, small detailed simulations of what he can do, which is much more detailed than what we can do on a much smaller length scale. But then on the other hand, what we get is like a bigger overview and a larger object that we can, so there's no free lunch. You have to find a compromise. So I think you could say it's close to nature, but it's of course coarse-grained. Yeah.
Okay, thank you. And one last question. How do you decide which experimental methods to use in general?
Okay, that's a very good question. So first of all, of course, we have a limited number and selection of methods available in our lab and with our collaborators. So those are the ones that we use. Sometimes we need something else that we don't have, and then we have to find someone to do it together with. This also mostly works well. And it's always good to first ask the question, what do you want to measure? What's your ideal outcome, and then think about which method you want to use. So it's interplay of availability, but then of course also what you like to see and what you would like to measure.
Okay, thank you so much for speaking. It was a pleasure to have you here. And now we're moving on to Yonit Maroudas-Sa on a talk about using the physics of liquid crystals. So looking forward to your talk. Thank you.
Thanks. Just tell me if you can hear and see me.
Yes.
See my screen? Great. Okay. So hi, everyone. Thank you for the opportunity to talk to you all. So my name is Yonit. I'm a PhD student in the Technion in Israel under the supervision of Professor Kinneret Keren and Professor Erez Brown. And I'm going to tell you about how we found that the physics of liquid crystals can help us understand morphogenesis in the regeneration of an aquatic animal, the Hydra. Okay, so first of all, morphogenesis is the emergence of functional patterns and forms in living organisms. So this is a prime example of pattern formation in nature. And if you think about it from a physics perspective, this is actually quite a hard problem, right? It involves extremely complex, robust self-organization in a noisy and far from equilibrium system. All right, so a very big question that remains is how do these robust and functional patterns emerge to produce viable living organisms under varying conditions? Now, in general, morphogenesis involves the integration of multiple different processes, biochemical and biophysical, with very extensive feedback between them. And as I said, it's really not well understood how all these different processes are coordinated and integrated across scales to form robust and functional outcomes. Now, a lot of the research, especially over recent years and since the advances in molecular biology have improved, focuses on the details, on elucidating the details of specific biochemical systems, etc. But again, approaching this from a physics perspective, we think that a lot can be gained from obtaining or using a coarse-grained approach and to obtain basically an effective description of the process. And this enables to integrate the many different processes that are going on during morphogenesis and requires to identify a relevant coarse-grained variable to use to describe what is happening and develop a predictive framework for understanding the dynamics using these variables. So, sorry. Yeah, okay. So for this purpose, we use a model organism, which is called the Hydra. So the Hydra is a small predatory freshwater animal. It's a relative of jellyfish and sea anemones, so it's quite basic. It's about 1 centimeter long and has a very simple body plan. So it's a hollow tube with the foot on one end and the head on the other, and tentacles that it uses to catch its prey. And this I'm emphasizing is a multicellular organism, and this hollow tube which forms its body is comprised of 2 cell layers which function both as skin cells and as muscle cells. Okay, now the really cool thing about the Hydra, what makes it an excellent system to study morphogenesis, the emergence of form, is its remarkable regeneration capabilities. So if you take a piece of tissue and cut it out of the main part of the body of a mature Hydra, this piece of tissue will fold into a hollow spheroid fairly quickly, within an hour or so. And in fact, within about 2 days, it will regenerate into a fully functional mature animal with a foot, a head, and tentacles, and everything. So I'm going to show you a movie of how this looks. So this movie is taken using under a microscope, and it looks like it's not playing. So let me just figure out why that. Okay, here we go. So you can see the initial tissue folds into a spheroid, and the process is moving around. And in this case, within even less than 2 days, you see already the emergence of the tentacles, and we end up with, although a small, fully functional, healthy, mature animal. Okay. So I told you that the cells that make up the 2 layers of the Hydra body are both skin cells and muscle cells. And if we look at the details of the organization of the muscle fibers in the Hydra, we see that the muscle fibers are very, very organized and very well aligned in a typical fashion. So what you see here is the muscle fibers and we use Hydra that are transgenic, so they're manipulated to express a fluorescent protein that attaches to the muscle fibers. So this way, using fluorescence microscopy, we can image and look at these muscle fibers. And you see the individual fibers are aligned really quite perfectly parallel to each other and parallel to the body axis. So these are fibers which are connected across multiple cells. You can see the size of the cell and the small outlines on the edges. So these are really across the whole animal. And these muscle fibers are comprised of the same components that comprise our muscle cells, and also that Sarah mentioned of the cell cytoskeleton, actin filaments, and myosin molecular motors. But we're not going to focus on these, on the details. What I would actually like to focus on is the organization of the fibers. So this type of parallel alignment, from a physics perspective, is called pneumatic alignment. So a pneumatic system is a system in which an objects align along a preferred orientation parallel to each other. And this orientation can be locally described by a director orientation field. So you might be most familiar with the term pneumatics from pneumatic liquid crystals, specifically maybe from optics, where elongated molecules align parallel to each other under specific conditions, can be light or temperature dependent, and according to the alignment can refract the light in different ways and are used, for example, in LCD screens. But also pneumatic systems are quite common in biology and have been studied quite increasing in being studied in recent years, for example, bacterial colonies, cell cultures, and in our case, the muscle fibers in the Hydra. So what we're going to see is why this description and why the analogy to other pneumatic systems helps us understand the morphogenesis process. Okay, before that, when we look at a pneumatic system, a very important feature is the possibility of the appearance of defects in the pneumatic alignment. So defects are singular points at which locally the parallel alignment is not maintained, and then they form typical patterns around this point. So these different types of defects are classified by what is called their topological charge, which is just the number of rotations of the director orientation field around the center of the defect. So what you can see on the left is 3 types of defects: the +½ the -½ and the +1. So again, you can look at the +½ and you see that the orientation field rotates exactly half a rotation around the center, and then the -½ is also half a rotation, but in the opposite direction, and the +1 is a full rotation. Now, in a closed system, the total charge of defects that are found in the system is constrained by the topology. So, for example, on a spheroid, no matter what the distribution of defects, the total charge together must equal +2. And again, the most familiar example of this is if you think of longitudinal lines on a globe, right? They can't be parallel throughout the whole, over the whole globe, and they meet at 2 points, the 2 poles, which are exactly these 2 +1 defects. So when we look at the Hydra, we see during regeneration and in the mature Hydra exactly these types of defects. So let me show you some examples. So during regeneration, when the tissue forms a spheroid, if we look at the muscle fibers, we see exactly these 3 types of defects. So the +½ that you see on the left, the -½ that you see in the center, and the +1 that you see on the right. So what you're seeing in these images here at the top are the microscopy images. So this is how it looks when we take images of the fluorescent signal coming from the muscle fibers. And below is our quantification of the fiber alignment. So the black lines, the orientation of the muscle fibers, and the color is what is called the pneumatic order parameter. So this is a mathematical measure of how parallel locally the lines are. And therefore red will indicate well parallel alignment, and the blue will indicate a place where the fibers are not parallel to each other, and this is the location of the defect. Okay, so if we look at the mature Hydra, it turns out that the location of the topological defect is extremely characteristic and robust. It will be like this in every single Hydra we will look at. Now, since topologically this is a closed surface like a sphere, so the total charge of the defect, if the full surface is pneumatically aligned, will exactly have a charge of +2. And this indeed is comprised of a +1 defect at the mouth, a +1 defect at the foot, and then also a +1 defect at the tip of each tentacle, which is balanced out by a pair of -½ defects at the base of each tentacle. Okay, so what happens during regeneration? So the first thing that we saw is that if we cut a piece of tissue from the body of the initial donor Hydra, initially this full array of well-ordered parallel aligned fibers is completely maintained in the excised piece of tissue. But then I told you that the next step of regeneration is that this open piece of tissue folds into a spheroid. So in that, it changes its topology. And initially what we end up with is a spheroid in which one side has a very well-aligned parallel array of fibers so that it's still inherited from the original Hydra. But then the spheroid will also have a large region which basically lacks ordered fibers. And this is where the edges of the tissue stretched to close and meet to form the spheroid. Then over roughly the first 24 hours, through a process of induction of order, parallel fibers will form also in the disordered region. But now these regions will also include defects due to the change of topology. Okay, so we've seen that initially in the open excised piece of tissue, the array of parallel aligned fibers is maintained. And then folding introduces defects. And we can follow these defects and we see that they are long-lived and we can follow them over time similar to regeneration times. And we've also seen that in the mature Hydra, the defect location is not random. It's very, very robust. And the defects appear exactly at the site of the morphological features. So this suggests that maybe if we can use the defects and the pneumatic description of the muscle fibers to build an effective description of the fiber dynamics. And maybe this can help us understand what is happening here in the regeneration process. Okay, so in order to do this, we follow the regeneration process using live microscopy. So I'm going to show you a movie here, which is the same spheroid from 4 different angles. Okay, so you can see the above in green, you can see the muscle fibers in the regenerating tissue spheroid. And below you can see, same as before, the black lines represent the orientation of the fibers and the color represents the pneumatic order parameter. So I'll remind you, red means well aligned and blue means either defect or disorder. Okay, so let's see how this looks. Okay, so again, it's a bit hard to follow when you're not used to it, but you can see initially there's some disordered regions and the fibers begin forming there. And you can see the emergence of defects. Actually, the images below help you identify where the defects are. And again, the process here is highly dynamic and there's definitely a lot going on. Okay, and you start seeing here the emergence of the +1 defect. You can see it now currently on the right. You start seeing the tentacles coming out. All right, and the defect at the foot as well. Okay, until we end up with the morphology of the mature Hydra. So you have the, sorry, that's not what I meant to do. Here, so you have the mouth, the tentacles, and the foot, as you saw before. Okay, so looking at many movies like this, we wanted to understand how the distribution of defects evolves over time. And again, if we can learn from this, how we get from the initial distribution of defects to the final distribution in the mature Hydra. So we counted and measured how the defects distribution evolves over time. And initially, okay, there are very few defects because the disordered region has not yet formed its aligned fibers. And within the first 24 hours or so, the full array of aligned fibers will form. And you can see that by far the most common type of defect at this 24-hour point is the +½ defects. And then what happens over the rest of the regeneration process is that the number of +½ defects reduces. These are the blue lines here with the diamonds, and the number of +1 defects increases, okay, until we reach the configuration which characterizes the mature Hydra, which is a +1 defect at the foot and a +1 defect at the mouth, but no +½ defects. And here we're not counting the defects associated with the tentacles because the number of tentacles varies, and we're focusing on the defects that define the body axis, which are the foot and the mouth. Okay, so how can we understand this? Where did these +½ defects disappear, and how do the +1 defects appear, and how do we get from the initial configuration to the final one? Okay, so it turns out if we look a bit closer at what's happening to these +½ defects, they move relative to the tissue. And the way that we can look at this is by fluorescently labeling a group of cells in a certain location in the tissue, and then looking at the location of these cells relative to the defect. And when we do that, we see that the +½ defects move large distances across the tissue compared to our labeled cells. And also they always move towards the round end and not in the opposite direction. Okay, so how does this help explain where they disappear to? Well, the most common thing that happens is that actually two +½ defects facing each other, even if they start off quite far from each other across the tissue, will move towards each other and end up merging and forming a +1 defect. The other option, which is slightly less common, is that a +½ defect and a -½ defect, if they're oriented correctly, the +½ defect will move towards the -½ and they will actually end up annihilating. So you end up with just a parallel array that contains no defect. Okay, so this helps us understand where the +½ defects are disappearing, but what happens to these ones that merge to form a +1 defect? Well, when we follow them and through to regeneration and see where they end up in the mature Hydra, these pairs of +½ defects that merge into a +1 end up almost exclusively becoming the site of the foot of the regenerated animal. Now, contrary to this, if we mark the cells underlying a +1 defect early on in the regeneration, these cells actually remain extremely stationary in relation to the defects, and it will be these same cells that are at the site of the +1 defect, which will become the site of the mouth of the regenerated animal. So although in the final animal, both the mouth and the foot have +1 defects, these originate very differently over regeneration. Okay, so let's focus on the +1 defects and specifically those forming the mouth. So we've seen that we can identify the site of the cells that will become the +1 defects very early on in regeneration, can be 4 hours even after the initial tissue excision. And I remind you, regeneration takes about 2 days. And these same cells will be the ones that will become the mouth of the mature animal. But I'd like to emphasize that the formation of the mouth is a significant biological process. It requires cell differentiation and many changes here. So what we'd like to understand is maybe something that is special about these +1 defects is helping to drive these changes. So if we look at the structure of think about the structure of the defects, I told you again that these are muscle fibers and they're contracting again and again and applying large forces. So the site of the +1 defect is in fact a focal point of these contractile fibers and the forces that they pull on the site. And therefore this focal point will be characterized by also weaker adhesion between the cells because you don't have fibers spanning across them, and also higher tension because of this pulling. Very strong evidence that we have for this is the opening of rupture holes at the site of the +1 defect. So I showed you, maybe you remember from the movie earlier, that the tissue during regeneration is highly dynamic and we have quite a lot of expansions and contractions. And during the contractions, there's often rupture holes that open in the tissue to relieve osmotic pressure. And when we looked at where these rupture holes are opening, early on when there are no well-defined defects, they can open in disordered regions. But once the defects are formed, they will open exclusively at the site of +1 defects and not ever in ordered regions. So this is a very strong indication that the mechanics of these regions is different and makes them much more liable to rupture at the defect sites compared to at other sites. So to kind of put all these things together, what we've learned about the +1 defects is that they appear very early on in regeneration. They remain stable in relation to the underlying cells. Their structure can provide a unique mechanical environment. And evidence for this is that rupture holes form at this site, and I don't have images here, but also we see that the dynamics of the cells and their shapes that they acquire at these sites are very different from the others. And that these sites of the early +1 defects almost exclusively become the site of the mouth of the regenerating hydra. So all these together lead us to suggest that these +1 defects act as organization centers for the formation of the head. And what is very interesting about this is that by looking at the organization of the fibers and the defects, we're actually able to identify the cells that will become the mouth much earlier than any known biochemical signals to date. So this really enables focusing on these special sites to understand the many different processes that have to feed back to each other in order to form the mouth. Okay, so to summarize, we've seen that the physics of pneumatic organization, similar to liquid crystals, provides a useful and predictive framework for characterizing the muscle fiber organization in regenerating hydra. And that the +1 defects that we can identify very early remain stable and act as as organization centers for the formation of the mouth specifically. And therefore, this brings us to suggest that by using the pneumatic orientation field, this provides us essentially with a coarse-grained biophysical field that definitely interacts with other mechanical and biochemical factors. But by looking at it, we're able to reliably predict and therefore understand the robust formation of the functional tissues in the regenerating hydra. Okay, so with this, I'd like to thank my supervisors and our group, and I'm very happy to answer any questions.
Thank you so much. That was such an interesting application. Okay, we have a couple of questions that we have time for now. So firstly, do all regenerated hydras look alike with the original body and among themselves? And how are the defects inherited?
Right. So the regenerated hydra, once they finished regeneration, can differ in the number of tentacles. But apart from that, they will have the same body plan. I will mention, and I didn't go into this at all, some hydra can, and therefore the name, can regenerate with multiple heads or multiple feet. And in that case, I actually have some images here I can show you. I'm sorry. I'll just reshare that a moment. I can show you the images. So these multi-axis animals, the same happens. You have an extra head, right? If you think about it, so you have an extra +1 defect, but then you have the pair of -½ defects at the junction, which exactly balance this out. And same, we can follow the -½ defects very early and identify where the junction will form in the two different heads.
Okay, thank you. What causes contractions?
Right, so these are muscle fibers. So I mean, the contractions are accompanied by calcium spikes and etc. It's not okay. So there are contractions that are coordinated across the tissue, also with neurons. The hydra does have a neural network, but not all. So it's actually not completely known how they are triggered and the different types of contractions, whether they depend on the neurons or not.
Okay, thank you. And one final one. How would this model be applied to other organisms?
Right, that's a very interesting question. So, I mean, the first of all, the system of actin fibers and myosin motors is ubiquitous. So these are really the main force, well, not main, but very common force generators in living organisms. And if you think of muscle fibers, so they appear in the Hydra, but they also appear, in our bodies. And if anyone has ever damaged a muscle or a ligament or something, and you're told to kind of apply a moderate force, right, to help align basically to help healing, what you're doing is you're applying force and helping to realign your fibers. So the idea that the fibers are both applying forces and then are also sensitive to the forces and aligning in accordance to that is very universal in biology, and that's the idea that maybe by looking at these things we can understand a more general concept.
Okay, great. Thank you so much. Okay, we're now going on to our final talk of the day. So John Devany is talking about physical constraints on cell growth and division. So welcome and thanks for coming.
All right, great. Thanks for the introduction, and thanks for the invitation to speak here. So today I'll be talking about physical constraints on cell growth. And so basically what we've been interested in is understanding in the context of tissues how cell growth and division occur. So on this slide I'm showing you an example where when we take cells, they'll organize into complex 3D structures. So here's an example of cells sort of forming the boundary of a spherical structure. And then here's the same fluorescence images of the same sorts of structures that we form in the lab. And then we do things like measure here we're labeling the cell membrane so that we can make 3D constructions of cells and measure their size in different contexts. So here you can see the same structure being formed out of cells of very different sizes. And then we can also use fluorescent methods to understand more about the state of the cells. So in this example on the right, cells which are labeled in pink here are undergoing DNA synthesis, while others are not. And so I'll show you a little bit about how we can sort of use these measurements of things like cell size, cell growth, and cell state to understand simple rules about how cells grow and divide in the context of tissues. And so the question we're trying to address here is understanding really how the size of multicellular tissues is controlled. And here I'm showing a couple of examples of embryonic development where basically we see two very different behaviors going on at the cell scale. So in the left, these are some developing frog embryos. And what we see is that they start with only a single very large cell, which undergoes these rapid cycles of division into smaller and smaller cells. And so the embryo remains about the same size, but you see a very large change in the number of cells. And so this large cell partitions into many small cells, which will then start to undergo 3D deformations to form the embryo. And then on the right, we have an example of a mouse embryo, which is made up already of many cells at this stage, but they're rapidly dividing and growing, which is increasing the size of this embryo. And so this growth and division occurs for a long time until at later stages, you start to see these 3D deformations, also caused by differences in growth as well as cell mechanics. And we wanted to understand more about how growth and division are regulated to give you these sorts of behaviors. And I'm going to focus in this talk on epithelium, which are a simple type of tissue which are found throughout the body. And their function is to serve as a barrier between the outside of the body and the inside of the body. So different examples are like kidney, intestine, or skin. And these often form a single layer of cells. Some of them are several layers of cells thick. And so they'll have a dimension in one direction, which is about the size of a cell, 10 microns. And then they're forming these large two-dimensional sheets that fill space at the millimeter scale. So they'll be extended in the other directions out to millimeters. And these tissues tile space, so there's basically a simple relationship then between the tissue size and the number of cells and their size, where the tissue size is just determined by the number of cells times their individual size. And then these two things, the number of cells and their size, are regulated by two independent processes. The number is determined by the regulation of cell division, so one cell dividing into two. And then the size of cells is really about the regulation of cell growth. So how a cell increases its size and the amount of mass over time. And so people have started to study these processes in much detail in a lot of different systems in about the last 10 years. And here I'm showing you a couple examples of single cells. So on the left, these are bacterial cells. And over time, they grow and increase their length until they've reached a specific size at which the bacteria will then divide into two. And then you see this process repeating itself again, each of these daughter cells growing to a larger size before dividing. And the same is true if you look at a single isolated mammalian cell. So here they're tracking the cell and it divides and then it goes through growth before another round of division. And if you look at the volume of the cell, it'll divide in half and then it will slowly regain its volume over the next cell cycle before dividing again. And so in this system, growth and division are coupled with one another through some molecular mechanisms which people have began to study. And the system grows exponentially until it sort of runs out of nutrients, which end up stalling growth and division. But things are much less clear in the context of tissues, where just like I showed you on the 2 slides previous, sometimes you can observe very different behaviors going on. Sometimes you see things like cleavage, where large cells just divide into smaller cells. And so you can get large changes in the number of cells that make up a tissue while remaining at the same volume. And then in other cases, like in mouse development, you get increases both in the number of cells and the size of the tissue. And the cells basically remain at a similar cell size. So here they've labeled some of the cells with a fluorescent protein so that they can monitor them over time. And what they see is that this group of labeled cells gives rise to more additional cells after 2 days. And you can see that this patch of cells has sort of increased. And this relationship between the cell's growth and division behavior and its context in the tissue has been appreciated for a long time. And it's been termed as contact inhibition. So this idea that contact of a cell with other cells in the tissue controls its proliferation. And it's also been appreciated that when this becomes dysregulated, it leads to dysfunction of the tissue in different examples, like in cancer, where cells grow much too rapidly. And then this causes the 3D architecture of the tissue to become distorted, which then leads to further issues like these cells migrating away from the tissue and invading other parts of the body. And so we wanted to sort of go back and think about whether or not we can that's a sort of qualitative description of what's going on. And we wanted to see if we could come up with a more quantitative understanding of this contact inhibition. And so in collaboration with 2 of our colleagues, Martin Falk and Arvind Murugan, and also UChicago, we developed a sort of simple framework for comparing growth of cells within tissues with their ability to grow in the absence of spatial constraints like this, in the absence of contact inhibition. And so the idea is fairly simple. We would look at our system of interest, so some tissue that we're interested in studying, and then we would see how it changes its size over time. So this function A of t could be something which just depends on the details of the specific tissue that we're interested in. And then we compare that with what we would expect single cells to be doing, which is growing exponentially, as I described to you a couple of slides ago, with some characteristic time, tau, at which the cells divide on average. And so in most cases, this might depend a bit on the cell type, but it's usually about 1 to 3 days. And then by comparing these 2 rates, we can sort of understand what the role of this tissue confinement or cell-cell contact is on controlling the cell growth. And so we have this metric we call the confinement, which basically describes the relationship between these 2 growth rates. And so when the confinement is 0, the 2 growth rates are the same. And so the cell growth is equal to what we would expect to see for single cells in isolation. And if the confinement is 1, that would be corresponding to a tissue growth rate of 0. And so spatial constraints are basically completely suppressing cells' ability to grow. And then we can basically quantify we can measure this quantity, and we can see like how close to being entirely confined are the cells. And so then we can start testing, whether this is a good model of understanding cell growth in tissues. We can make epithelial tissues in the lab. And so we can grow our epithelial cells in tissue culture using sort of standard methods. And then we can take these cells and we can make different types of systems with them. We can either make single cells and study their growth and division behavior, or we can make these epithelial sheets in vitro. And so here I'm showing the one of these tissue sections I showed a few slides ago of what these sort of structures look like in vivo. And then we can make similar structures in vitro, where you can see these cells in contact with one another. And so the interesting thing that we can also do when we make these tissues in the lab is we can sort of manipulate their geometry. And this actually allows us to also control the way they grow and expand. And so I'll be talking a bit about this model system we've been using that we call expanding monolayers, which we use as a model for tissue growth because they grow in an interesting non-exponential way. And basically the way we make this system is we start by putting our cells within a well that we fabricate out of a material called PDMS. So we can put cells into the sample just in a specific area, which is typically a millimeter scale area, and they'll form a nice monolayer within that area, and then there are no cells in the surrounding area. And we can remove this PDMS at a certain time, and then this tissue will start to expand out into the empty space that we've left behind. And what's interesting is that the cells do this in a very characteristic way. Basically, cells migrate at the edge of the tissue at a certain velocity. And so that this tissue, the yellow circle here shows where the cells were at an initial time. And then we're looking at the tissue 2 days later after cells have migrated out a certain distance, which is VT here. And what's interesting about these tissues, as I said, they grow non-exponentially. So for either a small or a large tissue, as shown here, over the course of 2 days, the radius of these two tissues increases by the same amount. And so what that means is for a small tissue, the area has increased by a very significant amount, whereas in the other case, it's increased by a much smaller amount. And so these expanding monolayers grow in this nice characteristic quadratic way. So the area is going like the velocity squared times time squared. And so that means that the area is only increasing like the square root of its size. Whereas what we would expect for single cells is an exponential growth where the area is increasing and proportional to the area. So like I described, we're comparing these growth rates and they become more and more different from one another as A becomes larger and larger. And so we can tune the size of the tissue by changing the sort of molds that we put the cells in initially to manipulate how much confinement there is. And I just want to emphasize, so this V here is really determined by this process of cell migration. And we can also make perturbations. I don't have time to show this data, but we can make perturbations to cell migration that affect how the tissue itself grows. And so then we want to go and look at how this tissue growth goes on to affect cell behaviors. So we can really zoom in here and look at individual cells and see how they're growing over time with some other function, small a of t. And like I said, the tissue is growing in this specific way, like the square root of its size. And if there is this feedback between cell and tissue growth, we would expect that the individual cells are also growing in proportion to the tissue area. And so I'll be showing some data where we start to measure cell growth within the tissue. And we're going to use a sort of trick using a fluorescent labeling method to measure this cell growth at, in a much larger scale. And what we do is we label the cells with a specific dye, which labels protein in the cell. And we do that at the beginning of the experiment. And then as the cell grows and divides, the dye will become diluted, so the concentration of the dye will decrease, and the intensity we see when we look at it will become smaller and smaller. And so you get this simple relationship that the intensity we measure is equal to the ratio of the initial volume to the final volume. And so it can tell us how much growth has occurred. So we label our cells, and we make this system with labeled cells. And then once we remove the barrier, the cells will, or the tissue will expand, and we can see the cells growing and diluting this dye. So here I'll show you the sort of first two limiting cases. So we know that there's going to be a big difference in growth between single cells and a large confined tissue that isn't able to undergo any growth. And that's what we see here in the experiment. We see that the dye has become very dim in this system made of single cells after we look 2 days later, compared to a large monolayer where almost no growth occurs. But then we look at these differently sized tissues, where by starting with a larger and larger tissue, we've sort of reduced the total amount of tissue growth that occurs. And then what we see is that the amount of dye dilution that we see is also changing as a function of the tissue size. So indeed suggesting that there's a feedback between the cell growth and the tissue growth that I was talking about in the last slide. Another interesting thing that we observe here is that the cell growth is quite uniform across the tissue. We didn't necessarily expect this to be the case. It might have been the case that the more migratory cells at the edge could be growing differently than the cells in the center, due to like differences in the sort of mechanical cues that they're feeling. But then when we go and quantify this data now across a large range of systems, we see that there's always this nice correspondence between the fold change in cell mass produced, so this change in intensity, the amount of new protein being synthesized by cells, and the change in the system size that we observe. So how much has the circle expanded in terms of its area? So we have this feedback between tissue and cell behavior that we can measure, and it's a quantitative effect. And so then we can go and what we've started to do is now plot different things as a function of this variable, the confinement. So we have this nice relationship between how much cell growth occurs and the amount of confinement. And then we've also started to look at different signaling pathways within the cell. So this is just an example of a signaling pathway that's thought to control cell growth in tissues. It's called IAP. And we see that the IAP signaling is changing as a function of confinement as well. So then we were interested in seeing what happens to cell division under confinement. And we found something pretty interesting to us, which is that the cell division didn't seem to depend on how much confinement there is. So here we're using a fluorescent marker, which can tell us which cells are preparing to divide and which cells are not dividing. So the cells labeled in pink here are synthesizing new DNA. And this can give us an idea of how many cells are going to divide as a function of time. And what we see in these differently sized monolayers is that the amount of division is basically about the same, whereas the amount of growth is being suppressed the larger the tissue becomes and the more confined it becomes. And so we get this decoupling between growth and division in the tissues. And what that means is that the cells, because they're undergoing division more quickly than growth, the cells are getting smaller. And so in a highly confined tissue, what we see is that a large cell divides into two smaller cells. And then through another round of division, the cell size can get even smaller. And we've done a lot of characterization of this behavior. And what we see is that the cells continue to divide until they reach a certain size where the cell cycle becomes arrested. So if we wait a very long time, cells all sort of approach the same final size. And the size is quite different than the size that we observe in single cells. And so we can summarize these effects of tissue confinement. This tissue, variable that depends on the tissue state, which is controlling cell growth through feedback. And then we also see this effect that the cell size reduces when division occurs more quickly than cell growth. And we wanted to ask a couple of questions related to this, like whether or not we think tissue confinement would be occurring in vivo systems inside of animals. And then we also wanted to know if we could think more about why the cells stop dividing at a specific size. And so I'll just show a couple of quick data that suggest the answers to those two questions. And so one of these is that when we make these tissues, we see this reduction in cell size across a variety of different types of cells. And so here we repeated the same sorts of experiments. I showed all the data I showed so far was in a specific type of kidney cell called MDCK, but we can also look in a type of intestinal cell or skin cell, and we see the same reduction occur in the cell size due to confinement. Then we looked across a bunch of data in the literature. So I was able to find measurements of the cell volume across 15 different epithelial tissues in vivo, so in the body, so from these tissue sections in the body. And then also 15 measurements of cell size made on single cells in cell culture. And we see this, there's consistently this difference in the cell size in these two different contexts, which matches what we observe in our system, suggesting that these epithelium in vivo are all feeling these effects of cell confinement. Then we wanted to understand, what sets this minimal size at which cells stop dividing. And one thing that we noticed is that as the cells are becoming very small, the concentration of DNA in the nucleus must start to get very high. So it's been seen before that as a cell gets smaller in size or larger in size, the nucleus also scales its size in proportion to the cell. So we see this correlation between cell size and nuclear size in our experiments. But then I've also highlighted two limits, two physical limits on this plot. So one is sort of obvious that the nucleus couldn't be larger than the cell, so this region is not allowed. But then there's another region here, which is not allowed because the nucleus would be too small to contain a full genome worth of DNA. So it's been seen before here. These are 3D reconstructions from TEM of chromosomes, and the volume occupied by a full set of chromosomes is about 90 cubic microns. And then likewise, you could also do a calculation of how much space DNA would occupy if it was fully condensed. So DNA wraps around these histone proteins, and if you just do sort of a simple back of the envelope calculation of how much volume this would take up, it's about 40 cubic microns. So this is a lower bound on the size that a cell can be. And so what we see is we see this bound here that the cells begin to approach. And then what we've done is we went and we made a perturbation to the cell cycle. So we force the cells to continue dividing even when they normally would not. And then that's these light purple points here. And what we see is that we can push the cells really close to these physical bounds. And then what we start to see is that then the nucleus becomes very large in the cells. So normally the nucleus takes up about a third of the volume of the cell. But once we make this, once we force the cells to continue dividing, the nucleus starts to become big and take up a lot of space in the cells. And then also what we see in those cells is we can label the cells for DNA damage. So we can see areas where the DNA is being broken by a staining method. And what we see is that when we force cells to become abnormally small, then they start to undergo a lot of DNA damage as well. And so just to summarize all of this, we've done a lot of work just looking at the behavior of cells within tissues, both to develop a quantitative framework for explaining this contact inhibition phenomenon, and then also understanding how cell size is regulated and the consequences of misregulation of cell size in tissues. So with that, I'd like to thank you for your attention. Thank the members of my group and Margaret, and also our collaborators on this work, Liam, Holt, Martin Falk, and Arvin Mergen. And I'll take some questions if there are any.
Thank you so much. It's amazing how many different applications we've had throughout this event. So yeah, one question. Out of the work that you've done, what has surprised you? And also, are there any implications or applications that could come out of this?
Yeah, so this effect actually surprised us a lot because there is this notion that people have been talking about a lot for the past 10 years and even beyond, that cell size is usually kept constant. So we always thought that in these, I did a lot of work before studying cell behavior in the monolayers, like cell migration, collective cell migration, and cell rearrangements and this stuff. And I always thought that the cell size was just something that sort of kept constant. And so we were really surprised when we started to see that you could get these like changes in cell size occurring like this. And then as far as applications, I think the main thing we're thinking about is in relation to certain types of cancers. So in cancer, the growth can become misregulated in the tissue, and that leads to overgrowth and 3D deformation of the tissue. So that's something that we would sort of predict here, is that if the cell growth is not being regulated properly, the tissue itself, that would cause it to become disturbed in structure. So we might look into that further. And then the other thing is related to another type of cancer called small cell cancers, where if the cells are similar to how we force the cells to divide more and become smaller. In this type of cancer, the cells, there's a mutation in the division regulation, and then the cells do become very small. So we wonder if that also leads to like what we saw, this DNA damage and cell dysfunction.
Okay, well, thank you. And thank you to everyone who came from IUPAB to speak for us today. It was a pleasure to have you all. So looking forward to seeing all of you tomorrow in the third and final day of our event. Before we finish, we're just going to have a brief presentation about IUPAB again. So I will just share those slides in 1 minute. But thank you so much for joining us.
So thank you very much, Ruhi. And let me reiterate your thanks for the speakers, Sarah, Yonit, and John. Fabulous talks. Perfectly pitched for a physics audience, and I hope it enthused some young students to consider biology and biophysics as a career. I'm just going to talk briefly about IUPAB because many people might not know what IUPAB is. Have you got the slide there, Ruhi? It's coming up.
Yes, it's loading. So
it's loading, good, thanks. And just to advertise tomorrow's event, Helmut Grubmuller will talk about theory and simulation in biology, and then we will have 2 other talks as well, one on simulation of nanopores, and then one also on transport systems across membranes from young scientists. So tomorrow's event will start at the same time. So IUPAB is the International Union for Pure and Applied Biophysics. The president is Manuel Prieto from Portugal. Our treasurer is Christina Zizon from Paris, and then Ron Clark in Sydney as our secretary general. We have an executive committee with 12 elected councillors worldwide. Next slide, please. Yes, thanks. So IUPAB is the World Federation of Organizations in Biophysics. We have about 61 countries with societies of adhering societies to IUPAB, and that constitutes around 12,000 individual members. That includes national biophysical societies, national research councils, and academies of science. There are also 3 regional organizations of biophysics: the Asia Biophysics Association, the European Biophysics Society Association, about 35 countries there, and then the Latin American Federation, all associated with IUPAB. Next slide, please. So IUPAB's participation within the International Science Council, and this is an overarching non-governmental organization of 40 international scientific unions, 140 or so national, regional scientific organizations, including all of the major academies around the world, and also advises the UN on global science policy. So we are part of this organization also. And there are sister organizations of IUPAB, for example, IUPAC for chemistry, for physics, for mathematics, for biological sciences, and biochemistry and molecular biology. So there are a number of these organizations to which many of you may or may not know that you also exist. Next slide, please. Our mission statement within IUPAB is to coordinate and support research and teaching in biophysics. And then our activities include, for example, our triennial congress that occurs internationally around the globe, often in parts of the world which are less accessible, but also in more developed countries. And then we support conferences, schools, and workshops. And we have three major task forces. This is an event under one of them, the education and capacity building, working with IAPS, for example, biochemistry and chemists. We have a structural biology and a big data in biophysics, and all of these organize events under the umbrella of IUPAB. Next slide, please. And just here are some of the IUPAB congresses and supported meetings coming up. For example, in India, we have a focused meeting in Canada coming up. There's no Biophysical Society in Southern Ireland, but this event is a joint event with the British Biophysical Society to get more interest in biophysics in Ireland. And then also there's going to be a course and a biophysics congress in Brazil towards the end of the year in September. And then our triennial congress, I mentioned, that's going to be in Kyoto the second time. The first time it was way back in the 1970s, will be in Kyoto in 2024. IUPAB travel bursaries for students up to €1,000 to attend these meetings. And we also support travel bursaries for those students who are not in eligible countries to gain support from FEBS and EMBO. And the last slide, please, just to say that we look forward to seeing you at meetings and hearing from you in IUPAB. So thank you, everybody, Zlatan in particular, for putting the event on and making sure it all worked, Ruhi and your other colleagues for chairing and so on, and the questions, and to all of the speakers. And we look forward to seeing tomorrow at 1500 UTC time. Thank you very much, everyone. Goodbye.